Hi ladies, this is a presentation about the stages of meiosis. So what is meiosis? Well, meiosis is the process by which the body produces gametes. And these gametes are what we call sex cells, so your egg and sperm. All the rest of the cells in your body are what we call somatic cells. So during meiosis, a diploid cell will divide twice and produce 4 haploid reproductive cells. Okay, these the egg and the sperm. Now I'm going to go into what haploid and diploid means. So diploid means you have 2 sets of chromosomes, or homologous chromosomes. When we say homologous chromosomes, we mean one from mom, one from dad. So when you're thinking about a specific gene you actually have 2 copies of that gene, okay? One that you got from mom, one that you got from dad. And we'll kind of get this understanding of diploid a little bit more when you actually do the lab on meiosis. Whenever we're talking about diploid cells, we refer to them as 2N, so that means they have 2 sets of chromosomes. Haploid means you have 1 set of chromosomes, or N, okay, just 1 set. And this means you'll either have 1 of those sets from mom or 1 of those sets from dad. And this will make a little bit more sense when we do our lab, but just for right now, make sure you get all these definitions down. Okay, so just like how mitosis works, meiosis, there's also interphase during the production of reproductive cells. So interphase just means that a cell is going to start with 2N, okay, so it's going to be a diploid cell, and it's going to go through the same phases that you see in mitosis. So G1, growth, S, DNA replication, G2, more growth. Okay, so pretty similar to what we've seen so far. Okay, so let's go on to the stages of meiosis. So we break meiosis down into meiosis 1 and meiosis 2. Meiosis 1 is going to be a little different from what we've seen before, but meiosis 2 is going to look a little bit familiar. It looks just like mitosis. So during meiosis 1, the main goal is to separate homologous chromosomes. So you're going to start with a diploid cell in interphase. So this is going to be interphase. Not sure why letters are disappearing. Okay, this is 2N. Then the cell is going to go into prophase. And this is prophase 1. Metaphase 1 is the next step. These stages should seem very familiar. Anaphase 1 and telophase 1. Okay, throughout this process, okay, we have homologous chromosomes here, so we're going to have a diploid cell, still have homologous chromosomes, diploid cell, still homologous chromosomes, diploid cell. When these cells actually finally split, okay, either moms or dads, they're going to be split up, so you're going to produce haploid cells. Okay, I'm going to go through each of the stages a little bit more in depth. So, prophase 1, the duplication of the chromosome. So the duplicated chromosomes will condense, just like we see in normal prophase. You're going to be able to see those chromosomes because all the DNA is now tightly wrapped. The chromosomes are going to find their homologous partners. So they're going to find either moms or dads. So this would be like moms right here, and this would be dads. They're going to have the same copy of a gene. So maybe this one has a copy for dimples on it, and this one has a copy for the gene for dimples as well. And what's going to happen is this very special process called crossing over. So homologous chromosomes, because they have the same genes on them, okay, they're about the same size, same genes, they can swap a little bit of DNA. So you can see here they're going to create these recombinant chromosomes where they swapped a little bit of DNA. This is really important to create some variation. Also during prophase 1, the nuclear envelope is going to disappear. During metaphase 1, the spindle fibers are going to attach to the centromeres. Homologous pairs are going to line up together in tetrads on the metaphase plate. And this is where the law of independent assortment is going to come into play. So this just means that homologous pairs will line up randomly. So they can line up in a variety of different ways. Okay, so let's look at this process. So right now, this is prophase 1. You can see the homologous chromosomes have found their partner, a little bit of crossing over taking place, nuclear membranes breaking down. Metaphase 1, lining up on the metaphase plate with their homologous chromosome. Okay, they line up independent of each other, so this set of chromosomes doesn't have to line up a specific way in relation to this set of chromosomes. Next is anaphase 1. So during anaphase 1, spindle fibers will pull homologous chromosomes apart. This is where they have the law of segregation take place. So this means that homologous chromosomes have to separate. Okay, so they have to be pulled apart. Okay, segregation, separate. And then chromosomes are going to be pulled to the opposite side of the cell. So let's look at this for a second. Okay, those homologous chromosomes are being pulled to the opposite sides of the cell by the spindle fibers. And then when we enter telophase, the nuclear envelope is going to form again, and you will have two separate cells. Okay, and this cell is now haploid, okay, because we've separated the set, so there's only one set of chromosomes in each. Okay, meiosis 2. So meiosis 2 is going to begin with those two haploid cells that we created. Okay, so this would be prophase 2. Okay, and this is a haploid cell. This is metaphase 2. Anaphase 2. And telophase 2. Okay. This whole time we're going to have haploid cells. We're just going to basically separate. The whole purpose of meiosis 2 is basically to separate our replicated chromosomes, just like mitosis. So meiosis 2 and mitosis are basically exactly the same process that you learned about. So during prophase 1, nuclear envelope is going to break down. You have your condensed chromosomes. During spindle fibers are going to start to attach. During metaphase 2, those chromosomes are going to, the replicated sister chromatids are going to be put on the metaphase plate. During anaphase 2, the spindle fibers are going to separate those sister chromatids or replicated chromosomes. And then you will, during telophase, new cells will form, okay, with cytokinesis, and you will have 4 haploid cells with unreplicated chromosomes. So let's go through this just to get your notes down. So meiosis 2, exactly like mitosis. Prophase 2, nuclear envelope is going to disappear. Chromosomes still have 2 sister chromatids attached by centromeres. And then the spindle fibers are going to attach. Okay, metaphase 2, sister chromatids are on the metaphase plate. Anaphase 2, sister chromatids are going to separate. And telophase 2, cytokinesis is going to finalize the 4 haploid gametes. You'll have nuclear membranes reform, and you are pretty much good to go. So this is the process of meiosis. So meiosis is going to differ a little bit between males and females. So in males, we have the process called spermatogenesis. Okay, you can see. Let me get my pen. Okay, making sperm. This is going to start at puberty. At the end of meiosis 1, you'll have 2 haploid cells. At the end of meiosis 2, you'll have 4 haploid sperm. Okay, so 4 sperm right here, and these little guys are all haploid. All 4 are going to survive. They'll all be the same size. They will be genetically different, though. So part of the purpose of meiosis is to create genetically different cells. So they'll all have a different combination of mom and dad's chromosomes. Females are a little bit different. Meiosis 1 is going to take place in the womb. So this is when two haploid cells are going to be created. There'll be unequal cell division. Each month, Meiosis 2 divides those again unequally, making one kind of like stronger egg and 3 weaklings. These little weaklings, we call them polar bodies. And basically, what's going to happen is that only one egg will be produced. Okay, and you'll have these polar bodies. Okay, they're all haploid, but the reason that there's unequal cell division is because this egg needs to have all kinds of things inside it in order to help a growing embryo. So the mother kind of sets up this one egg with all this nutrients and proteins and all kinds of things that growing embryo would need. Okay, basically, the sperm is just carrying chromosomes. It really doesn't have anything else other than the genetic material to bring. So the female has to have all this other things that an embryo will need, okay, until in humans this embryo will actually implant in the mother's uterine lining and be able to develop. Okay, so that is meiosis and spermatogenesis and oogenesis there are 2 additional questions on your note sheet. You do not need to answer those for tomorrow. Just bring this completed note sheet with you, and we will go through the process of meiosis and walk through it a little bit more in depth. Okay, please bring any questions.